Understanding meiosis part 2, the process. So, as with any type of cell division, meiosis starts with interphase, and in interphase, the DNA will replicate to form complete chromosomes. So, 2 sister chromatids attached by a centromere, as explained in the previous video. Once interphase has happened, then the cell will proceed into prophase 1. In prophase 1, what will happen is the nuclear envelope will disappear, a spindle apparatus will form, and the homologous pairs of chromosomes will attach to the spindle apparatus, as shown in this diagram. It is worth mentioning at this point that this is the point where independent assortment occurs. See the next video for more details on that. After prophase, we proceed into metaphase 1, where the pairs of chromosomes will align at the equator of the cell. Then you proceed into anaphase 1, where the homologous pairs separate, and one from each pair goes to the pole of the cell. And then in telophase 1, the nuclear envelope will reform. Were this mitosis, the cell would now proceed into interphase, but as it is meiosis, the cell does not go into interphase, and instead it proceeds directly into prophase 2. In prophase 2, a second spindle fibre forms, this time at 90 degrees to the first spindle fibre, and the individual chromosomes that were separated in meiosis 1, those individual chromosomes, now attach to the spindle fibre. In metaphase 2, those individual chromosomes align on the equatorial plate of the cell. And then in anaphase 2, those individual chromosomes separate into chromatids, and they go to each of the poles. And then in telophase 2, you have your 4 diploid cells. Now, these cells do undergo interphase, and so those individual chromatids will replicate to become individual chromosomes, and that will still result in the formation of 4 haploid cells, because each cell will contain 1 individual chromosome from each homologous pair, ready to then fuse with another gamete to form a diploid cell. And that is the process of meiosis.